In this short video, I'm going to talk about genetic drift. Now, genetic drift is another way in which evolution can proceed that is not via natural selection. So previously, I'm going to write natural selection up here. And I'm going to say we're not going to go by natural selection. We're going to evolve by drift. Now, genetic drift. So might as well write that in there. Now, how exactly does that work? Let's take a look first at our gene pool here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 individuals. And I've made them all heterozygotes so we can see that the frequency, in this case, of big A is equal to 0.5, and the frequency of little a is equal to 0.5. Now, let's say that we're going to hold a special case where there is no selection allowed, all right, in our little model here. And this is a population that basically doesn't have any particular kind of conditions that are selecting on it. Or maybe there's no variance in the sort of ways that selection is acting, so it doesn't have any effect. But we can look at it like this, and we say no selection. We're also going to say that the population, in our case, is very small. Now, what we would expect under natural, in most cases, is that if the fitness of each of these individuals is equivalent, then everyone should reproduce equally. And in meiosis, for example, in the part we call independent assortment, each individual has an equal probability of handing off a big A or a little a. So you have a 0.5 probability and a 0.5 probability of handing off each of those, right? Now remember, as you've already gone through, I'm sure, the meiosis videos, you know that these two alleles basically spin around in space and only one can be handed off to the egg or the sperm. So we would expect, on average, in the next population, we would expect to have the same frequency, such that the frequency of big A should be equal to 0.5 and the frequency of little a should be equal to 0.5. But there is a problem, and that is there are only 10 individuals in this population. And so just like when we flip a coin, each time we expect an even probability of big A's and little a's when each one goes through, right, and makes their contribution to the next generation. But half the time we should get big A and half the time we should get little a, but we'll get deviations from that. So in small populations, we'll get those deviations as a result of the small sample size. In small populations, we get deviations from the expected frequency alleles in the next generation. Now, what that basically means is that if we flip a coin to see which of these guys will give so we'll take it out, you get a heads or a tails, right? You take this one out, you get a heads or a tail to see whether it gives a big A and a little a. About half the time, they'll give a big A. About half the time, they'll get a little a. But not exactly, because we only have a sample size of 10. Just like if you flip a coin 10 times, you should get 5 heads, 5 tails, but there's actually a pretty good chance that you won't get 5 heads and 5 tails, that you'll get 7 and 3, or 6 and 4, and 4 and 6, and 3, 7, that type of a thing. So this deviation from expected means that allele frequencies are changing. They change across generations due to sampling variance. Now, the sampling variance, as we talked about in lab, are these deviations from expected frequencies that occur due to small sample sizes. Now, if there were 1,000,000 individuals, we would get pretty close to 50% A and big A and 50% little a. But we don't have 1,000,000, we have 10. So this does some interesting things. If we plot this as the frequency of allele in time, so let's put this as 0.5, 0, and 1, and this is going to be, say, the frequency of big A, and this is generations. What we should see is when we have a very large population size, so I'll write infinite population size, and there's no selection, what we should get is that over time we should stay right here at exactly, right, 0.5, 0.5, 0.5. So always get expected. That's because we have the law of large numbers here, right? We always get 0.5. If, however, we make that same drawing for a very small population, from 0 to 1, we start at 0.5 in the middle. If we start at a frequency of 0.5 and say we have a population of 10 individuals, we don't know in the first generation, the frequency of big A could drop or go up. We don't know. So we'll go like this, then it goes like this, and then it could go like this, and then this, and then this, and then this, and this, and then this, and then this. And we get a whole bunch of noise on here. So this is random sampling variance. Now the direction of the change in each generation is not predictable, but the magnitude is dependent upon how small the population is. Smaller populations make more variance, and larger populations have less variance. And that's always what we see up here. Large population gets very little variance, and a small population, look at all that noise all over the place, and we could eventually lose alleles here, even though they're not under selection. So something important about genetic drift it does cause allele frequencies change, but we're not actually getting adaptation, right? So let's say it does not result in adaptation. And what that means is the alleles that are being favored here are not being favored because they help the organism provide a higher fitness in some environment or other, but in fact they just happen to increase or decrease in their frequencies. So genetic drift can be a way in which really good alleles that would be good for fitness, can be lost, and sometimes those are not good for fitness can increase. But it's really quite random. So this is a pretty fascinating way in which evolution does occur, but not due to natural selection. Those are the two big pathways, natural selection and genetic drift.